Here we are going to learn about viral diseases, which is an illness that's driven by viral infection. Today our focus is on adenoviruses, which is a virus that causes upper respiratory tract and also eye infection. It's not only infecting children but also infecting adults. Let's look at the structure of virus. So for this virus, adenoviruses is belonging to this Baltimore class 1, and as we know that most of the virus, their structure is very simple. They consist of only the genetic material and also the protein code. So this is same thing happen to these adenoviruses. For adenoviruses, is belonging to this family Adenoviridae. So the genetic material is a double-stranded DNA, which is in linear form with 26 to 30 kilobase. And this virus, it has only a naked isocosaheral capsid, which means that this virus, it do not have a lipid envelope. It consists only of protein outer layer, which is this one. And this is the diameter of virus, and with 11 capsid protein. And this virus, it can further divide into 4 genera depending on the target which they are going to infect. So that's why it's divided into 4 genera. And so far until today, we have a 51 non-human serotype, the adenoviruses that infecting human. For these adenoviruses, as you notice that the outer structure is having a knob fiber protruding from each of the 12 vertices. This is the fiber protruding. So this protruding fiber, they are constructed from the hexon and also the pentone bases. So for the fiber with the terminal knobs, they are project from the pentone bases. So this fiber are used to make contact with the cellular receptor protein to initiate infection, which means the adenovirus is going to use this fiber to interact with the host receptor, which is, for example, in human, so that they can be enter into the cell cytoplasm via endocytosis. So which host receptor they are interacting? They are interacting with the CAR receptor on the surface of the host, and the other secondary receptor interacting with the host is the integrin. This integrin protein is also found in the host cell. Once they interact with both receptors, then they enter via endocytosis. So for the adenoviruses, they are mainly infecting this polarized epithelial cell. What is the polarized epithelial cell? The picture here showing a non-polarized epithelial cell in human. Well, this is a polarized epithelial cell. So the target is only focused on the polarized epithelial cell, as the structure is easier for the interaction with the receptor. This virus, they survive for extended period on the environmental surfaces. So that's why normally the infection route is through contact, direct contact. So since this virus is purely only a genetic material and capsid protein, it do not have the lipid envelope. By this, is resistant to lipid disinfectants. However, it can be inactivated by heat, formaldehyde, or bleach. Now, let's look at the life cycle of the virus. So first of all, as you can see, this is the adenovirus. The entry of adenovirus into the host cell is via the interaction with the CAR receptor on the host cell. So the CAR receptor stands for the Coxsackie virus and adenovirus receptor. Once they interact with the host, what is happening is now they will enter via endocytosis, and then the interaction with also the secondary integrin receptor. Later on, what is happening is this virus will start to uncoat and then to release its double-stranded DNA into the host nucleus. From there, the virus will start to do transcription to produce its mRNA. Once they produce the mRNA, the mRNA will be released to the cytoplasm to start to synthesize the viral protein. At the same time, the viral double-stranded DNA will also be synthesized in the nucleus. Later on, this capsid protein synthesized in the host cytoplasm will be again imported into the nucleus and then assembled together with the viral double-stranded DNA. From there, they start to assemble to form a new virus and then escape from the host. So it just takes about 24 to 36 hours to complete the whole cycle. Now let's look at how this adenovirus, they can activate the DNA replication in the host so that they can start to synthesize their genetic material. This slide is showing the explanation. However, I'm going to explain to you using this picture. So first of all, if you look at this picture here, in a normal scenario, this is in a normal scenario, when the normal host cell, they are not in a replicating cycle, what is happening is, there's a type of protein. This protein is called RB protein. This is the full term of the RB protein. So this RB protein in the host, it will bind to the E2F protein, and then at the same time also HDAC protein. By this, it can repress the host transcription, which means at this point in a normal scenario, when there is no viral infection, what is happening is, when RB bind to E2F and HDAC, then your body cell not going to initiate transcription. And then, as we know, we have cell cycle, right? So in the cell cycle, during the S phase, the cellular CDK will bind to the RB. Once CDK bind to the RB, so the E2F will be released. The releasing of E2F will initiate transcription in the host. This is what happening in normal human's body. However, now, how this adenovirus, they can initiate DNA replication and also transcription in the host. In this adenovirus, the adenovirus is having this E1A protein. This E1A protein will bind to the RB. So once it binds to the RB, it's just like hijacking the RB protein. Once it hijacking the RB protein, the E2F will be released. So the presence of E2F will initiate transcription. By this, transcription and replication will be happening in the host. So it can force the host cell to do the viral protein. If you look at the genome, this adenovirus, the genome is covalently linked to the viral code with terminal protein. This is the terminal protein. And if you look at the replication cycle, first of all, this viral, the pre-terminal and polymerase protein initiate DNA replication. So the pre-terminal protein that contain a serine residue. What is happening is, it provides an OH group for the DNA extension. So there's no RNA primer are needed in this stage. Then they start to do replication. And you look carefully, there's no replication fork needed for the replication of viral DNA. And then only one daughter strand is provided at each round. Later on, the single strand DNA will circularize via the inverted terminal repeat. Then they start to form a panhandle structure with short double strand stem. From there, the replication cycle will take on. For this adenovirus, primarily is infect children, and then other certain serotypes also associated with other diseases like respiratory infection and also gastrointestinal infection. In terms of the pathology, so most children are infected with adenoviruses early in life without resulting in disease. For the symptomatic illness is depending on the type of virus. Some example like type 2, the patient may develop this stuffy nose and cough, while in older children they have pharyngitis. So and the type 3 and type 7, they may cause pneumonia in children, which is in an infant, which is less than 2 years old. For the type 4 and type 7 is the one that easily cause outbreak via respiratory tract infection. However, it may cause severe infection in those immunocompromised patients. Besides that, adenoviruses also can cause other symptoms like pharyngitis and conjunctivitis. So normally the outbreak is via the swimming pool, and it also can cause gastrointestinal infection. So the symptom infection occur mainly in infant only. So in terms of eye infection, so it's highly contagious and it can be the spread, the transmission normally is via contact with tears or the discharge. So sometimes some patients have this eye infection with adenoviruses, so they keep on touching the eye and the tear. From there, it can be transmitted to other patients. So in terms of for this endemic and epidemic respiratory infection, mostly infecting children in the early life. So normally it's less than 5 years old. So less than half of infection result in the disease. So the symptoms normally like mild, they are including the stuffy nose, cough, and pharyngitis. Some younger than 2 years likely they may develop this pneumonia. So most of the adenovirus infection, they are self-limited, which means sometimes the disease may subside themselves without any treatment. So all the 51 serotypes are endemic in the community. So for the seasonal variation, sometimes there's outbreak in the summer and also the winter may be slightly in different area. In terms of diagnosis, so for the viral diagnosis normally is done via serology. So we use the ELISA or antigen antibody interaction to identify the infection. So the viral isolation is from the clinical sample, either from the eye swab or from the conjunctival swab and other location. Further on, it can be culture. So once you culture them, normally we observe for this cytopathic effect. However, most of the most common method for most of the viral infection diagnosis is via this antigen antibody interaction. So we detect the presence of adenovirus IgM in patient serum. In terms of treatment, generally, unlike this bacterial infection, most of the viral infection we do not have antiviral. Only certain viral infection that we have this antiviral. So there's no specific treatment for patients with mild adenoviral infection. So for immunocompromised patient, we can give this antiviral drug. So antiviral therapy is generally reserved for patients with any form of immunocompromise. So this is one of the antiviral that can be used. So it inhibits the viral DNA polymerase and viral replication. So by this, it can suppress the replication cycle of the virus.